All right. Hi, everyone. It is 7 p.m., so I want to get started. I want to start by being the first to officially welcome you to Wednesday evenings at the Genome 2023. Yay! My name is Allison Feeder. I'm an assistant professor in the department, and it's my great honor today to get to introduce Dr. Gail Jarvik. Dr. Jarvik is I have a lot of titles to read, so I'm getting my sheet of paper. Arno G. Miltulski Endowed Chair in Medicine at the head of the Division of Medical Genetics, a professor of medicine and of genome sciences, an adjunct professor of epidemiology, an affiliate member of the Fred Hutch Cancer Center, and also a practicing physician. So lots of titles. Dr. Jarvik got her PhD at the University of Michigan in human genetics and her MD at the University of Iowa. She moved on to Penn for her residency, go Quakers, and here to UW for her fellowship, where she ended up being recruited to stay on as a faculty member. During her career, Dr. Jarvik has made enormous contributions to the understanding of complex diseases. So many people may be familiar with the idea of a Mendelian trait or a Mendelian disease, in which you think of the allele at a specific location being associated with a disease phenotype. And knowing that you have this allele may help you in terms of seeking different behavior or treatment or early interventions. Unfortunately, the majority of diseases are not Mendelian diseases. They're these complex diseases in which you have lots of contributions from many different genetic contributions hidden all across the genome, and this makes them much harder to study and disentangle and diagnose. And complex diseases represent some of the most important and challenging diseases, things like risk for stroke and heart disease and cancer and dementia. So Dr. Jarvik's approach is using these large clinical cohorts where we have information both about genetics and also about health outcomes, and putting these together to try to disentangle the contributions of disease and design ways to diagnose these things better. So in addition to these contributions to genetics and medicine, Dr. Jarvik also does really important work in bioethics, asking questions like, what do we do if we run a genetic test on someone and we don't completely understand the result? What do we do with that information? It's a hard thing to try to grapple with. So for Dr. Jarvik has received numerous accolades for all of her contributions. I'm not going to list everything because we don't have time, but she's a Pew Scholar in Biomedical Sciences, a lifetime National Associate of the National Academies, and was designated as a local legend from Washington. You'll have to ask her at coffee afterwards what that means. She's a preeminent leader in the genetics community, serving as the chair of an NIH study section, so helping the government decide what research to fund. And in 2021, she was elected as the president of the American Society for Human Genetics. So we're very lucky to have her today. Will you all join me in please welcoming Dr. Gail Jarvik?
Thank you so much. Appreciate everyone coming on this beautiful day. I'm going to take a little poll and say, raise your hand if you are not a scientist. Not a scientist. Okay. Now, keep those up. Further, add your hand if you are not a geneticist, but are a scientist. Okay, good. I just want to know who I'm talking to. So I am from a big family in which I never finished a sentence for my entire 18 years that I lived at home. So please interrupt if you have questions. This is a great size for conversation. And I will shoot things over the head of the non-science people, like that's guaranteed. So stop me and ask me to explain what I'm talking about. Okay, all right. So so I'm going to do a little show and tell from Allison. I'm going to talk first about Mendelian diseases, the more simply inherited, sometimes single gene. And then I'll move to complex diseases later in the talk. All right. And we don't want to forward. Tech person is running to my aid. There we go. I don't know. Just walk in the room and it fixes itself. Thank you. Okay, so I'm going to tell you a little about medical genetics here at UW because most of your speakers are not clinical medical geneticists. So the Division of Medical Genetics is a division of medicine. And so we're a little bit different because many, but not most, oddly, of our faculty care for patients with genetic disorders. So this is a early slide. We were founded in '57 by this man here, Arno Miltulski. This is like old school clinic conference. They sat the patient down, everyone sat around and interrogated them. We don't do this exactly this way anymore. We have 26 faculty. We tend to be on the more translational side of genetic research. We have an adult medical genetics clinic. We actually have one of the largest adult clinics in the country. We see 5,000 patients at UWMC approximately a year. We see an additional 2,500 at Fred Hutch. Those are cancer genetics patients. We see an equal number of cancer genetic patients here. So cancer genetics, breast cancer genetics, colon cancer genetics, things like that are our top disorders in medical genetics clinic, but we get a lot of cardiac genetics, probably 25% of our business, neurogenetics, connective tissue, adult biochemical, general adult genetics, lots of super interesting and rare things in this clinic. We have trained physicians in medical genetics for more than 50 years. We have a training grant that I think is like 47 years old now. People have occasionally said to me, like, can you send me the original training grant that you sent in? Because I'm going to write a new program. And I'm like, yeah, that's not going to help you at all if I could find it. Anyway, and then we started a genetics counseling program just a couple of years ago. Our first class graduated. It's a 2-year master's program. First class graduated this spring. So that was exciting. All right, so I'm going to talk about 3 different programs, one of them just extremely briefly. The first is the undiagnosed disease program, and that is like rare diseases. So this is part of a national network. They feed people into a national coordinating center, but by region, they are assigned to be seen and worked up. These are people with long diagnostic histories, but no one's figured out really what they have. The second thing I'm going to talk about moves to complex disease, and that is the eMERGE project, where we're looking at risk scores for common diseases like stroke and heart disease. And we're doing that again in UW patients as part of a national network. And then finally, I'm just going to mention the precision medicine cohort, which is a really exciting cohort that you all can sign up for if you're interested, and why it's important a little later. Okay, excellent. So this is sort of the assignment map of which patients go where. So we're up here in green. There we are, UW and Seattle Children's. So it's a joint project with the Children's Hospital. We take both children and adults who have these rare conditions. We have Alaska, part of us. We don't have Wyoming for historical reasons, but we just tell them to ask for us and they can get us. But we have, for those of you who don't know, there's like a whammy medical region in the Pacific Northwest that includes Wyoming. So we consider it our territory, even if the national center doesn't. So what's the goal? To end the diagnostic odyssey for patients. And there's some really interesting science. I'm going to go through one patient from our UDN in a little bit. It's a super complicated patient, so I'm hoping you'll just bear with me as I get through it. But the point of it is that it's a super complicated patient. So I think that all of you will understand that part of it. All right, so who can come to the UDN? You have to be a month old, you have to be medically stable and be able to transfer, and you have to have a condition which does not have a diagnosis after a reasonably thorough workup. So it's not really like, well, there's 6 more tests we'd like to do, let's send them to the UDN. It really is like they've done clinically what they should do and they still don't have an answer. People apply to the UDN, the majority of them are not accepted to the program. There are criteria for acceptance which are related to basically our ability to solve the case. If we don't think we can solve it, then we don't take it. You do not need to speak English to apply to this program. Okay, so the UDN does things that we don't do in clinic. And this is really the tension of the UDN, because in clinical medicine, we are limited, honestly, by what insurance will pay for. Okay, so there's a lot of genetics tools and other tools that we cannot throw at a patient's problem without extra research funding. So sadly, one of those tools is deeply going through the medical record. So we can have, 10 years of thousands of pages of medical record that needs to be summarized. This does unfortunately not happen in clinical medicine, right? Because you don't get paid for that in clinical medicine. So the doctors will skim through records, but to comprehensively go through the records is sadly a research activity. I wish it wasn't, but that's the story. So we can do more, I'm gonna have to learn to use this pointer. We can do more detailed DNA and RNA sequencing than you will find paid for by most insurance. We can get paid for genomes on children a little more often these days. On adults, it's super hard. RNA analysis, much harder. And other rarer things, even harder still. And then finally, you can do a variety of things, either specific therapeutic trials or model organisms. Again, would never be covered by clinical medicine. All right, so this is the network. We joined the network after they had already been there 5 years. So we joined now 5 years ago. And so this is the network numbers, applications received, 6,500, still working. So it's still an ongoing thing. So this is how many people are in the queue. Accepted, this is about 2/3. We accept fewer actually of our percentage, 2,000 evaluated and 676 diagnosed. I'll be honest to say that the diagnostic rate for this network is dropping now because people are coming through the door with a genome, which wasn't true 10 years ago. Okay, so historically, 81% of the diagnoses were from sequencing. And so that's obviously dropping. It is interesting and important that we learn more about what the genome's doing every day. And so we can take a genome that was generated 5 years ago, rerun it through our analyses pipeline and find the condition that was not even known 5 years ago. So reanalysis is a key step for people who have existing genomic data that hasn't been solved. RNA sequencing has been very helpful as well. I'm going to mention that later with my clinical case. Some of the diagnoses are straight up clinical. Our rate is actually about 10%. About 10% of the records we review, we can make a diagnosis without any further anything, just by having gone through those medical records and, in fairness, with a giant team of people to argue about what the patient might have, okay? But, again, I think it's the frustrating thing about medicine, like all the data's there. If you had it all together in a coherent way in front of the right person, you would have an answer already. And honestly, for us, that 10% is like overall of everyone we review. That's about how many people end up with diagnoses. That's out of maybe 100 accepted. I think I have that later, but it's interesting that we're, equally contributing by just reviewing records. All right. So the network as a whole has described 53 new conditions, like new disorders, like we found it, we checked it in a model organism. We believe that this genetic change is causing the problem in this person. Okay, so this is just the genetic analysis pipeline, which I am definitely not going through, but just to say that Liz Blue is a superstar and she's running the genomic pipeline and helps us find the things, because there's a lot of variation in your genome. She helps us pull out what could be interesting, what could be related to the patient's disorder. All right, so we're going to do a case. So this little cutie came to us, and I do want to credit the lead clinician for the unit is Katrina Dipple. She's the head clinical geneticist at Seattle Children's, the genetic counselor's Emerald. And then we had a bunch of genetic analyses that you'll see. Andrew Sturgachis' lab did the majority, Liz and Peter helping. And then the referring clinicians from a genetics clinic are here. Okay, so she's 3. I love it that they labeled her here. She's 3 months here, 9 months here, and I'm not sure of this, but it looks intermediate to me. So anyway, so we're looking at this little face. Does this look like a normal face? No, this is not a normal face. I'm not a dysmorphologist. I'm an adult geneticist. So even though I had to take the boards on this, I'm not great at it. But even I can see that there are some issues with this face. So I can see that the eyes are wide set apart. Do we see that? The bridge of the nose is actually quite flat. It's a little hard to tell depending on the angle, but you can see it there. The nose itself is small. Harder to see in this picture is that the jaw is quite small. And so this child had respiratory problems at birth because of a small jaw, her tongue obstructing her airway. She has another feature here. I don't see my clinical people who already know this case. If you are the parent of a child who was ever 9 months old, were they leaning against something when you took their picture at 9 months old? No, they were not. At 9 months old, you should sit up. This child cannot sit up at 9 months old. She can't support her head at all at 9 months. Okay, so that is another thing we can see in the picture. All right, so we're going to summarize her. So she had problems since birth. We make slides like this. Like this is a summary slide. I just pulled it off our internet and took off the identifiers. But this is the way we kind of keep the cases in our head because we have more than 100 of these. She had bilateral retinoblastomas, so tumors in her eyes, in the retinas of her eyes, both eyes. Okay, she had an abnormal structure of her brain, hearing loss, both ears. She couldn't feed and required a feeding tube. She is very hypotonic or floppy, okay, and that's why she can't support her head. She has these dysmorphic or unusual facial features that we talked about. She had one elevated biochemical assay. I'm not going to talk very much about that actually. Her growth is below. So here, I think most of us have seen standard growth charts that they put your kids on. You can see that her growth this is her weight, this is her length. So below, really kind of off the chart low. Her head circumference is a little bit better, but not normal. So she's got global developmental delays, and we can organize it by organ system, but I've said most of these things here. One of the things that we do is we translate every condition that we see into what are called human phenotype ontology or HPO terms, because that's the way databases are set up. So we're going to put this child into a database with this description, and we need to use these standard terms. Okay. So she had a clinical workup that I will show you, but the only thing it showed was a apparently balanced translocation between chromosome X and chromosome 13. Okay, we're going to come back to that. All right, anything else exciting on that slide? I don't think so. Okay, important. Her mother had a normal karyotype, and we got her dad's, she came through the door with that. We got dad, and his is also normal. Okay, so this translocation that she has could have been inherited from a parent. It was not. If it had been inherited from a normal parent, we'd feel a lot better about it, right? But it was not inherited from a normal parent. It is new in this child. All right, so then we have some plans for analyses. All right, so she came through the door with now, you're not going to know what all these tests are, but just believe me when I say this is a very thorough genetic workup. So they looked at her chromosomes grossly and everything was there, first test. They ruled out common genetic disorders that are associated with poor feeding and poor tone in children. All right, they did a comprehensive 230 gene panel of all the known neuromuscular disease in children. That was completely normal as well. They did a karyotype and a FISH for the RB gene. I'll come back to RB. And that's where they find this apparently balanced, strong apparently here, translocation. The retinoblastoma gene is the only known cause for retinoblastomas genetically. They happen sporadically. These are the eye tumors. She has bilateral tumors that would be extremely consistent with having a genetic change in this gene, and it's a recessive, so you need 2 genetic changes in that gene usually. So they looked very closely at that gene. They didn't find anything wrong with that gene, and it is on chromosome 13 notably, which is one of the impacted chromosomes, but way far away from where the actual break is. Okay, so they're working their way through it, and they said, okay, well, what if the fact that that 13 is now sitting on an X, and if you're born with 2 X's, one of those gets turned off in every cell, because if you're born with an XY, you only have 1 X. So somehow we have to dosage compensate in people born with 2 X's. So they said, what if it looks like it's turned off disproportionately? And those studies were also normal. And then they did some RNA sequencing saying maybe we're missing what's wrong with this retinoblastoma gene, found nothing. All right. So here's her karyotype. This is very old school. I love to see a karyotype because honestly, we do a lot of these things by array, but this is one of the times where her array to look at all the parts of the chromosome, they were all there. This is the way to find out that they're not in the place we expect them. So here's the normal chromosome 13. You can see this is not normal. And here's the normal X, and there's this extra chunk here that belongs over there. Okay. But as far as they can tell, cytogenetically, everything's there, just something's moved. Okay. All right. So no answer there. So they managed to get a trio exome. That's all the coding regions of the genome. And they do the kid and both parents. And they're looking for new things that occurred in the child that the parents didn't have. There's a bunch of stuff. There's always a bunch of stuff. None of it, trust me, is informative. They redid the whole thing with a genome, instead getting all those non-coding regions too, as well as looking at the mitochondria, because mitochondrial disorders, that's your energy of your cell. Those are associated with kind of floppy children with hearing problems and feeding problems. So mitochondrial disorder might have fit this child, but all completely normal. So, in essence, they also did, neuroimaging, muscle biopsy, a whole bunch of other biochemical tests. Lots of specialists saw this child. So this was a very thorough clinical workup. This is really like best case scenario for what comes into our program. And, all they find here was this balanced translocation, and then the, I didn't highlight this, but the retinoblastoma RNA, so that's like what's going to turn into the protein, is in the low range of normal, but low normal, not abnormal. All right, so I'm not going to walk you through the technology here, but only to say that Andrew Sturgachis, one of our junior faculty, who's amazing in genome sciences and medical genetics, has developed a technique to do many things at once in the genome. And so he and his wonderful lab ran this child through the pipeline that gives us genomic data, regulatory data, and then data about the RNA as well. Not going to tell you the details. All right, so here's where I need you to hold on with me. I promise we're going to get there, okay? So I'll go back one. All right, so we're going to look, this is the maternal chromosome 13 and the maternal chromosome X. They're normal. So now we know that the event comes from the father, okay? So this event could have happened either early, it happened on the paternal, haplotype, could have happened in spermatogenesis, could have happened very, very early in fetal development. Okay, so we have, these breakpoints. And the question really is that Andrew's asking, did it break through anything important right there? So we don't see a chunk of anything missing, but what was severed in those breakpoints? So he maps it all out and finds the fact that the word first is here tells you there's more, right? The first Mendelian condition that this child has is that they have a disruption of this gene, NBEA. The breakpoint goes right through the gene and causes it to create an RNA that's too short and gets decayed away. And so we're missing functional product from that chromosome for that gene. So that's one thing. That gene we know is associated with developmental delays. All right, so that's one. All right. Then, thing two, there's a second problem connecting this gene at the same break with this part of this gene. So you end up with this, what we call a fusion transcript. Okay, so it's exactly at the same spot. You ruined another protein. okay? You can see, for those of you who are interested, that it's the minority of the transcripts that they pick up have this abnormal protein. However, they used an assay to determine that this construct will cause an increased amount of this protein. Okay, so it's a gain of function there. All right. I should say, you can tell the slide game went way up in these slides. Andrew made these slides for me. It's very nice. All right. So finally, what's the last part of it? We still haven't explained the retinoblastomas, right? And remember, there was that X inactivation study that showed that the X should be pretty balanced. That shouldn't explain it, but in fact, I'm not going to walk you through this because it's very complicated. But the key is that when in fact Andrew looks at this, I'm going to go back. He can show from the X inactivation that the maternal haplotype, which is the normal RB1 gene, is basically turned off. So in 97% of the cells, the maternal gene is silenced and the paternal one is inactive, which causes you to be at risk for retinoblastoma. So I'm just, yeah, that's the bottom line here is 97% maternal is inactivated. And then you have this abnormal expression from the paternal side. All right, so I'm gonna skip over that and just say, okay, what does this explain in our sweet little patient? So this gene is disrupted, cut, and then won't make a protein. And so that we believe accounts for the neurodevelopmental delay. This fusion is in fact in pyruvate dehydrogenase, which is mitochondrial. So we talked about how they were suspicious for mitochondrial disorder, and in fact, that is what happened. And so that should account for the poor tone, the feeding issues, the growth problems, the hearing loss, and maybe the dysmorphic features. That's the part I don't have the best explanation for, but mitochondrial disorders sometimes have dysmorphic features. And then the retinoblastomas we explained by the X inactivation. So, all right. We talk about this a lot in medicine. I don't know if other fields talk about this at all. Occam's razor, plurality should not be posited without necessity. That fundamentally means try and find only one explanation for what's going on here. Okay, so lots of times, you know, you'll be trying to explain something on the ward and the attending will say to you, well, Occam's razor, it's probably just one thing. Less often, interestingly, people bring up Hickham's dictum, which is the patients can have as many diseases as they damn well please. So it turns out in medicine, however, so Hickam was an MD and Occam was a philosopher. It turns out in medicine, the patients can have more than one disease. And that is what we see a lot in the UDN. Now, honestly, for this little gal, I don't know if I want to, I mean, she has numerous things that happen, but it happened because of one event. So is that one disease? Is that three diseases? I don't really know. I don't have an answer for that. But many times we will have a person with many features and we will find an explanation for 2/3 of them, half of them, not all of them. There's two possibilities there. One is that we have expanded the phenotype. We've actually found someone who that genetic change has more features associated with, but probably more often, possibly, they have something else too. They have a second condition. Sometimes in this program, we have found the second condition as well. But that's one of the complications for us. And I think people get to us because they seem to have like way too much going on. So it's not surprising that it's more than one thing. All right. Okay, so these are diagnoses that we've made. This was through May because we wrote a report in May, so that's where I stopped. We did solve another one last week, but anyway, so, and you can see, because everything that has like little letters, like these are genes. So you can see a lot of this is genetic. So we're still solving a lot of these genetically. Some of them this is environmental. So this is someone who took an antibiotic, which unfortunately caused muscle disease. This is someone who had, chronic vomiting from cannabis, which turns out it happens. And by the way, smoking cannabis to help your nausea in this case does not help. But you can see they're very one-offy, right? They're very one-off, like everything's different. Like these are very, very rare cases for the most part. I should have made a slide of the ones that we could make the diagnosis without seeing, but I don't think I did. So, all right, so what have we done? So we, had about 300 applications. We're still reviewing 34 of them. We accepted 134 people. 96 have been evaluated, and about a quarter of those get a final diagnosis. And then, as I said before, we reviewed, 258, and 25 we could solve by medical record review. So that's where we are, behind where we wanted to be because we got funded and then COVID hit. So we're slowed down, but we're making up progress now. All right, so anyone can apply to this program in the world, even not just nationally, but in the world. If you're out of the country, you have to get yourself into the country to be seen, but you can directly enroll there. And so I'm always encouraging people who have friends and family members to apply to this program because it's unfortunately a resource that's going away because the funding ends in about a year. And then what happens to these people, right? That's really up in the air. I'll be happy to pontificate about that later. So this is a great group. These are the docs who see the patients. This is sort of the staff, but also Katrina's the other PI with me. She's the pediatrics PI, I'm the adult PI. And then Liz, as I said, does the analysis, but our genetic counselors are here and our study staff and lab staff there. And these are the people who do all the work, right? Because I showed you a big list of people before without me on it. All right, so questions about the UDN, because I'm going to completely change topics now. Yeah.
You mentioned that some of the phenotypes makes a portion of them. Is there any time that you got to go back and have to learn more about the genome, rerun all that data, or is it just that they're too complex and too associated that's not going to clarify information?
Such a great question. Right now, everyone that we don't completely solve, we rerun those analyses at 6 to 12 month intervals, right? So later, but when the funding goes away, I don't know what's going to happen to these. So, but right now we do go and look back. The other thing is all of these people are put into these databases that exist to match rare conditions, and we follow up. We could have put people together with other cases and sometimes 3 and 4, because once you find something, then everyone starts to find it. And so you can put those patients together and get a more broad idea of what the phenotypic spectrum could be for a specific condition. Yeah. Yeah.
Can you do anything for her?
Can I do anything for her? That is such a great question. I don't know yet. So the one thing that is possible is if the mitochondrial disorder can be addressed somehow biochemically. And so that I mean, this is like hot off the presses. So that's one possibility, but the answer may well be no. For the people in this program who get a diagnosis, about a quarter of them actually have a medical management change directed at their disorder. A number of them have changes in their care, but that could be physical therapy, occupational therapy. Sometimes, honestly, it is not continuing to care for someone who doesn't have a prognosis. That's a change in care. There have been N of 1 biochemical treatments developed for children in this program. So you're the only person with this treatment. Then some, so these are frankly resourced families who raise money and get companies to work on a treatment for this one child. That has happened in this program. So this child, I don't know. The other big impact for most of these is reproductive information for the parents. What is the risk of having another child? Because until we saw her, they didn't even know what it was. Like was it the translocation or not? It was unclear because many translocations cause no phenotype at all. So yeah, so we'd love to help her, but yeah. And frankly, when I started this program, I was surprised that 25% of the time we could actually make a difference. All right. Yeah. A little louder for me.
Why transportation part of the homes?
Why it happens? So it turns out this stuff happens all the time. Like translocations are super common. Just it's, the chromosomes, they line up, they sort. So if they break and reconnect to the wrong one, there's recombination events. This is very common. These sorts of mechanisms are very common. And the key for something that looks balanced is it disease associated or not? And that's if it's not seen in the family. Now I have seen families with 4 generations of a balanced translocation with no symptoms, but if it unbalances, so if you don't get that short 13 and that long X together in the same cell, then you're either going to have too much 13, too little 13, too much X, too little X, right? That's called unbalanced. And then those kids will have abnormal features. And so we have families who have generations of a balanced, they're fine, but the eggs or the sperm are not fine, and then affected children are born. Yeah. So other questions? Yeah. I don't know if I should say this as a clinician, but field development is super complex, genetics is super complex, and the fact that it works as often as it does is really remarkable. It really is. Okay, so now we're going to talk about common diseases, okay? So this is another study we're doing at UW. The idea here is to help people predict risk of common diseases. So I'm going to show you the list in a minute. And the way we do that is to take established research on genetic associations with those diseases, kind of put them all into a pot and say, well, if all of these variants make a tiny change in your risk for this disease, if we put them all together, does that give us enough information to tell you something useful? Okay, and that's called a polygenic risk score. And it can be hundreds of variants, thousands of variants, or millions of variants even in these scores. All right. So we are, this is 10 sites nationally, of which we're one, generating polygenic risk scores for 10 conditions. So that's polygenic. That's lots of little things. That's polygenic. We are adding them together with family history, clinical information, monogenic. So these are the Mendelian things, like the breast cancer gene would be one of these. And then social determinants, smoking, things like that. And putting them all together to a risk score and giving this information back to our participants. What is the point of this study? The point of this study is to see if it changes medical care. Because if it doesn't change medical care, if you don't do anything differently when you have this information, then believe me, insurance companies are not going to pay for it. Okay, so this is the network. And so you can see the black dots are the clinical sites. So here we are sitting all alone on the whole West Coast because this is a lab, that's not a clinical site. I happened to be coming back from an emerge meeting like 2 weeks ago, and I sat next to on the plane Pramila Jayapal, and I was trying to explain, she asked me what I was doing, like where I was coming from. So I said I was coming from NIH, and that, we're out here in Seattle, the only ones in this giant consortium representing our region and our patients. And she was like, what about Stanford? No Stanford. So anyway, we try and represent here. So this is the network. There are 10 sites. We are all throwing 2,500 people into a big analysis. And the reason we do these nationally, not just because it's hard to get 25,000 people at one site, but it has to work in different healthcare systems. Like if it just works in one healthcare system, it doesn't mean it's going to work in another healthcare system. So we really try this across multiple sites to see if we can consistently get improved care. All right, so the thing about polygenic risk scores, they're very different than like the breast cancer gene, where you have like a 50 to 80% chance of breast cancer if you have abnormal change in that gene. In this case, even when we add all this information together, we're generally talking about odds ratios of like, you have a 2-fold increase of the disease. You have maybe a 4-fold increase of the disease, but these are, small risks. They're common diseases, which helps. So if women have a 10% chance of breast cancer, then double that is 20%, right? But it's not 80%. Okay, so, but we don't really have a lot we can do to predict risk of common diseases, except for the people who have the very rare bigger genetic changes that are Mendelian. All right, so it's, family history is about the same thing. Family history for some of these conditions is 2- and 3-fold risk. Like if you have a first-degree relative affected, especially at a young age, you're already, but what is surprising to me is when you put them all together in one big regression equation, I kind of thought if you had family history in, the polygenic risk score wasn't going to help you very much, but they're surprisingly independent, which is something I thought a lot about. And it won't take a lot of time now, but it is surprising. The one, so in a consortium, you win a few and you lose a few. And what I lost was the reports read that you are high risk for the condition with a 2-fold risk. I don't consider that a high risk. I really did not want them to use high risk. Elevated risk was what I proposed. I was outvoted. So it's reported as high risk, but every one of these persons sees a genetic counselor, and the first thing the genetic counselor is going to say is, by high risk, we actually mean. All right, so besides predicting disease risk with these scores, another thing that you can do, and this is someone else's work, published paper, but I really like this, is like, okay, everybody over here that has a high risk of heart disease, if you put them on a statin, you decrease their risk of heart disease. Yay, that's what we all know, right? But if you had a high polygenic risk score and I put you on a statin, you get a much bigger bang for your buck, right? And so a big question in clinical medicine, and I'm an internist, is like, well, my cholesterol's about this high, I don't smoke, I'm not overweight, I am male, which is a risk factor, should I be on a statin or not? And this is one potential tool to help us decide which of those borderline cases should be on a statin. So, all right, so these are the conditions we're working with. So 3 heart conditions: atrial fibrillation, high cholesterol levels, clogged arteries, which you have to see there's a correlation between high cholesterol and clogged arteries, chronic kidney disease, breast cancer, prostate cancer, colon cancer, diabetes, and obesity. In children, we are doing asthma and type 1 diabetes. At our site, we are doing mostly adults, 2,400 adults, only 100 children. So we will not be doing very much of that. So most of these have a polygenic risk score associated. Colon cancer does not. And that's because the polygenic risk score for colon cancer was not yet good enough in people of African ancestry. So your ancestry matters for these scores. I'll show you more on that later. But for colon cancer, was not ready for prime time. But we are also doing what we call monogenic testing, or these Mendelian tests. So there's some rare high cholesterol things, heart disease, breast cancer. But if you add these things all together, that's about 3% of the population. And so what we didn't want to do is say, oh, you are not at high risk for this and miss this. So even though this isn't really part of our experiment, we want to make sure that these 3% of people know that they are actually at higher risk than these people are. And so that's why we're doing the monogenics there. All right, and so these are the genes. This is really for the geneticists. They're the standard, breast cancer, Lynch syndrome, and familial hyperlipidemia. And like I said, about 3% of these are going to have positives. Everyone who has a positive Mendelian result or a high-risk PRS has the opportunity to see a study genetic counselor for free. All right, so this is our cohort. We're trying very hard to have a diverse cohort. We are trying for 2,400 adults. And we are trying to get 75% underserved. So underserved can be non-European ancestry, it can be rural, it can be LGBTQ, it can be disabled. So there's a variety of definitions for underserved here in this study. If we break down our ancestry so far, so we're closing in on 2,000 people, we have about a third European ancestry. Some of these are Hispanic because these are not mutually exclusive categories. You could check multiple things here. But this is only gonna get worse. And by worse, I mean more European. And why is that? Because I have tapped out inviting to the study every diverse patient at the University of Washington. Literally, we've invited about 50,000 people. All right, so we worked very hard at including LGBTQ individuals, which are very underserved in medical research. And that's been successful pretty far. We didn't work as hard on self-identified disabled, but we have something there. This, you will see female at birth, 64%, always in genetic studies. The women, female at birth, over enrolled. We've invited equally as many XY as XX individuals, but this is who actually enrolls. It may be because of interest in the breast cancer genes that may drive some of it, but we do know, and there's data to support, that in families, it's the mothers, it's the grandmothers that hold sort of the genetic information. It is not unusual to do genetic counseling for a couple and have the wife be able to tell you her husband's family history of disease better than he can. There's data to support this. This is not just my personal observation. Okay, so why diversity? So this is kind of a hard plot, but the general theme of it is there's a lot of Europeans in our genetic studies. And in fact, as you go from 2006 to 2018, it got worse. We're seeing less representation. I mean, there's more studies overall, but the proportion of Europeans is not doing great here. Why is this? Because the biggest data set available for these studies is the UK Biobank, the United Kingdom Biobank. And it is mostly European ancestry. And so that is where you can go to do your association study, but there's not diverse data. So one of the things that we're trying to do in the All of Us program, which I'm going to talk about in a minute, is improve this and make data available that's more diverse. I hinted at this before, that if you have a polygenic risk score that you've developed in people of European ancestry, it doesn't work as well in other populations, and particularly African populations. I'm not going to go into why, but it's important that you can't just take this score developed in European ancestry and apply it to other ancestries. And in fact, a direct-to-consumer company withdrew a test that had that problem. All right, so the All of Us National Precision Medicine Cohort, the goal is to collect over 1 million people. They will have very deep medical records. They will have wearables for some of them. They will have extra testing, and they will have genomic data, genomes, and these arrays that look at all the parts of the chromosome, but not in detail, done for each individual. We are one of the 3 sequencing centers, genome centers for this program. And the goal of the program really is to add diversity. So at least 50% of the participants will be ancestrally diverse in this program. And I think they're at like 650,000 right now. Oh yeah, there we go. I put it on. How about that? So yeah, so we're getting them in there. We've got, this is how much genomic data we have so far. The genomic data got delayed because of like FDA and IRB things, which are interesting, but not worth our time tonight. Okay, so when we look at the polygenic risk scores we're planning on returning, this is just a couple of them. For example, you can see here's an odds ratio of 1.73 in Europeans. That's the predictive power in Europeans, but it's weaker in Africans. If you look at the confidence intervals, not clear how different it is, but consistently weaker in African ancestry individuals. So this is something that we need to explain to our patient participants, right? That if they are not of European ancestry, their odds of having disease is not as high as someone who is of European ancestry for many, but not all of the conditions. All right, so this is just what the report looks like. So they get a list of everything we looked at. And then this is a positive report for type 2 diabetes. And so it says you have this higher risk, but it also says like, here are the healthcare recommendations that we have, because we know that physicians are not going to know what to do with these reports. Right. I think the best message of physicians is treat it like even more family history. Like that's kind of the care recommendations, but we want to be very drilled down and say, these are the things that you should be doing. And these are the things the study will be looking to see, did they actually do? All right, so one of the things, and again, this is high risk, the upper 2% is considered high risk. That means if you're at the 97th percentile, right, you are getting the normal report. the not high risk report, it's called. So the fact that you are not high risk does not mean you are low risk for these things. In a perfect world, right, you would just tell people you're at 50th percentile risk, you're at 5th, you're at 95th percentile risk, but in a clinical study, you need an outcome. They got it, they didn't get it. They're high risk, they're not. If you give people numbers, the math is really problematic to actually look for outcomes. Did it change care, right? So that's just one of the realities of clinical research. All right, so for all of the conditions, there are things that we do, things that we measure, things, risk factors, and then there's patient education for all of them as well. As I said, the goal is to see for the people who got a high risk report, pooled over all the conditions with adults and children separately, were healthcare actions more frequent in the high risk versus the not high risk people? Okay. With the people who got low risk being a control group, really, there are going to be lots of people, about 25% of the people will be high risk for one condition. So 75% will not be for any condition. They're the controls. Okay, privacy, very important. So we're doing genomics on people in a clinical context. All the identifiers remain local. So the name, the patient number, the address, the zip code, the date they were seen, that all remains local. But we are sharing de-identified data both across the consortium because the analysis is going to pool all this data together. And also they will be posted in an NIH repository because by the rules of NIH, if they give you money to do genomic research, you have to share that data. It's required, which is a good rule. But there are certainly potential participants who don't want their data shared, so then they can't enroll. New this cycle, because this is actually the 4th cycle of eMERGE. You might have seen the eMERGE 4, a couple places on the slides. These cycles have been 4 to 5 years. We're in cycle 4, so this program's been going on a long time. We are no longer sharing this information nationally. Pregnancy terminations, gender transitions and dysphoria, and child abuse. We used to. And now we are in a political environment where we don't feel like we can share these data. That's not safe for our participants. Okay, so you too, if you get your care at UW, you can enroll in the study. So if you get your care at UW, you can email me, you can email eMERGE 4, I'll just forward your email to them, and enroll in the study. And so we will finish enrolling hopefully by October-ish, and then be returning results generating data, returning results for like another 6 months. And then we'll see if it all worked. All right, so conclusions. UDN, reanalysis of negative results is important. Thank you for asking that question. Many people have more than one disorder. This is not clinical care. The program ends, the research money goes away. How do we take care of these patients? What happens to these patients? Our patients have been very aggressive in campaigning to Congress. They got us an extra year of funding, but the federal government has this idea that this is clinical care and that we should be doing this clinically, and it just simply is wrong. All right, so eMERGE, polygenic risk scores, these are coming. You're gonna see these. You'll be offered these in direct to consumer context. Executive physicals sometimes already offer these. You really have to be sensitive to the ancestry. You have to develop it in multiple ancestries. And actually, if you start by developing it in one ancestry and add a second ancestry, that improves the prediction for the first ancestry. Okay, so it's really best for everyone if you have all the ancestries in. They're weak predictors, but they are predictors. And then finally, we need more diverse data, and we need more diverse scientists as well. Data sharing is critically important. UW should be very proud of its history of data sharing. We're really on the cutting edge of let's just share the data with everyone and let's throw it on a website. And the field has really done that. These are the eMERGE team people. David Crossland is my co-PI. These are the people talking to all the participants, and you can see it's a big team. We've got the informatics people, the database people, the lab people, lots of doctors. So that's my lab with a couple other, there's Liz Blue who does the analyses, they're faculty. Katrina's my other PI for UDN, and Clem has been in all of my lab pictures for 25 years. He's one of our senior faculty. There's a story behind that, but that we could do that over wine. Anyway, questions about eMERGE, common diseases. Yeah.
So the example for with the statin, regarding like multiple, I guess my question is regarding like multiple diseases in the same thing. And particular treatment that would help those diseases. Is the conclusion that people with a higher cardiac risk score tend to be tend to have see more progress with whatever treatment, just based on the fact that those diseases might be the same family?
So the statin, that is a targeted treatment for high risk of heart disease. So these people are known to have high risk of heart disease. A statin is going to, you'll see that gap, and the statin will be more effective in people who are obese, who are diabetic, who have any known cardiac risk factor. You'll see that gap. So that slide was simply showing that the prediction is good enough that this gives us enough information and new information about the risk of disease that we can better target statins to people there. But it's like any other risk factor. If you have the more risk factors you have, the better off you are on a statin. And this is just one more of them. Did that answer the question? Okay, great. Other common disease questions. So common disease research, which is what I spend most of my time doing, is just job security. Like these are really hard disorders to chip away at, but so important because this is what people die from in this country, right? I'm an internist, I'm an adult doctor. These are the patients that I see. So that's sort of why I'm passionate about common diseases. Okay, other questions? Any question? Yeah.
Yes, you mentioned the reanalysis of the negatives. And you also mentioned that you're starting a study, you don't want to give a non-binary yes or no answer for the high risk. When you do that reanalysis, do you think you can provide more of a spectrum of where essentially you are?
Yeah, so for this study, no. For eMERGE study, they're just going to be told they're in this top or they're not in this top, because we're going to be following out for years and years. Okay, so we will not be going back to these people and saying, actually your risk was X. For breast cancer, I didn't really go into this. For breast cancer, we give people like very detailed 10-year risks because that's the standard for the field. So even though they're told your polygenic risk score is high or not high, they are actually given their exact 10-year risk of breast cancer because those models are already available. So where we can and where it is a clinical norm, we will give people that information. Yeah, but no, I'm really quibbling with the terminology high risk, really, like that's my quibble. It's not, it's elevated risk. It's not high. I mean, we all have different definitions of high, and I had a patient who had I think it was like 1 in 350 chance of their child being affected with something, and she thought that was enormously high. And this was, a family that wouldn't consider a termination. And so it really was like, do they roll the dice or not? And they didn't, at least in, I don't, subsequent to me seeing them, maybe they had, but their decision was 1 in 350 is too high. 5% of births have some significant abnormality, right? So to me, 1 in 350 is kind of negligible, and you're taking that 5% roll every time you have a baby, right? But people don't think about that. And it is really like your perception, but I don't personally want to label it as high. Yeah, other questions? I think we have refreshments. Thank you very much.